Breakthroughs redefining biology, from same-sex procreation to age reversal in mice. The boundaries of biology are being pushed in ways once thought impossible, with scientific advancements opening doors to new possibilities in reproduction, aging, and beyond. From enabling same-sex couples to have biological children to accidentally reversing aging in mice, these discoveries challenge our understanding of life itself. Below, we explore some of the most intriguing recent breakthroughs, including the revolutionary O chromosome concept, age reversal in mice, and other cutting-edge developments reshaping science. The O chromosome, a new path for same-sex procreation. One of the most provocative advancements in reproductive science is the concept of creating biological offspring for same-sex couples using an engineered approach to chromosomes. While the specific O chromosome referenced here appears to be a hypothetical or emerging idea not yet fully documented in mainstream scientific literature, it aligns closely with ongoing research in vitro gametogenesis, IVG, a technique that could allow same-sex couples to have children genetically related to both parents. The process described involves removing the nucleus, containing the genetic material, from a donor ovum, replacing it with the nucleus of a somatic cell from a male partner, and then fertilizing the modified egg with sperm from the other male partner. This results in a zygote with genetic contributions from both individuals, potentially producing a male child with a unique chromosomal configuration, referred to here as an O chromosome, capable of reproduction. While the term O chromosome is not standard in current genetic nomenclature, it may represent a novel designation for an engineered sex chromosome or a placeholder for a modified genetic structure. Real-world research supports the feasibility of such techniques. Scientists like Mitinori Saito at Kyoto University have made significant strides in IVG, successfully creating functional eggs and sperm from mouse skin cells using induced pluripotent stem, IPS cells. In 2023, Saito's team reported progress in generating primitive human eggs from IPS cells derived from blood, hinting at the potential for human applications. For same-sex male couples, this could mean converting one partner's skin cells into an egg, which is then fertilized by the other partner's sperm. For female couples, creating sperm from female cells is more complex due to the absence of Y chromosome genes, but breakthroughs in mice, such as producing offspring from two female parents, suggest it's not far-fetched. The implications are profound. IVG could eliminate the need for donor eggs or sperm, allowing same-sex couples to have children who share their DNA. However, ethical concerns loom large. Bioethicists like Henry Greeley at Stanford warn of risks, such as unintended genetic mutations or the potential for designer babies, if combined with gene editing tools like CRISPR. The technology is still years away from safe human use, with challenges like ensuring proper meiosis, chromosome halving, in lab-created gametes. Yet the prospect of rewriting reproduction is closer than ever. Accidental age reversal in mice, a diabetes drug surprise. In a serendipitous discovery, researchers studying diabetes and blood pressure medications stumbled upon a potential fountain of youth, at least for mice. In 2023, a study revealed that a combination of drugs, including metformin, commonly used for type 2 diabetes, and valsartan, a blood pressure medication, not only managed these conditions but also reversed epigenetic markers of aging in mice. These markers, found in DNA methylation patterns, are like biological clocks that indicate an organism's age at the molecular level. The experiment began as an investigation into how these drugs affect metabolic health. However, when researchers analyzed the mice's tissues, they found that treated mice exhibited methylation patterns typical of younger animals, effectively reversing their genetic age. This wasn't just a cosmetic change. Treated mice showed improved organ function, enhanced muscle regeneration, and extended lifespans compared to untreated controls. The drugs appeared to reset cellular pathways linked to aging, such as those involving sirtuins and AMPK, which regulate energy and stress responses. While the exact mechanisms are still under investigation, the discovery suggests that existing medications could be repurposed to combat aging. Human trials are a distant goal, as translating mouse results to humans is notoriously tricky. Aging is complex, influenced by genetics, lifestyle, and environment, and drugs like metformin carry side effects. Still, this accidental finding has sparked excitement about geroscience, the study of aging as a treatable condition, and could lead to therapies that extend healthy lifespans. Note, specific details on this study are based on emerging reports. Further primary sources are needed for confirmation. Other groundbreaking scientific advancements. Beyond reproduction and aging, scientists are making leaps in diverse fields, each with the potential to transform lives. Here are 3 additional advancements that stand out: CRISPR babies and ethical frontiers. In 2018, Chinese scientist He Jiankui shocked the world by announcing the birth of twin girls, whose embryos were edited using CRISPR to disable the CCR5 gene, aiming to confer HIV resistance. While the experiment was widely condemned for its ethical violations, lacking transparency and introducing unknown risks, it demonstrated the power of gene editing in human embryos. The edited genes could be passed to future generations, raising both hope for eradicating genetic diseases and fear of slippery slopes toward genetic enhancement. He was sentenced to 3 years in prison, but his work underscored the need for global regulations on germline editing. Ongoing research now focuses on safer, more precise CRISPR applications, such as treating sickle cell anemia in adults, with promising results. Brain-computer interfaces. Merging mind and machine companies like Neuralink, founded by Elon Musk, are advancing brain-computer interfaces, BCIs, that allow direct communication between the brain and external devices. In 2024, Neuralink reported its first human implant, enabling a paralyzed patient to control a computer cursor and play chess using only their thoughts. BCIs work by recording neural signals and translating them into commands for devices, with applications for restoring movement, treating neurological disorders, or even enhancing cognitive abilities. Challenges remain, including ensuring long-term implant safety and scaling the technology, but BCIs could redefine human capability, raising questions about privacy and identity. Note, specific Neuralink updates are based on recent reports. Primary sources may vary. Synthetic embryos from stem cells. In 2022, researchers at the Weizmann Institute in Israel created synthetic mouse embryos from stem cells without using sperm or eggs. These embryos developed heartbeats, neural tubes, and other structures, surviving for up to 8 days outside a womb. While not fully viable, they mimic early embryonic development, offering a window into how life forms. This could revolutionize developmental biology, improve infertility treatments, and reduce reliance on animal testing. For humans, synthetic embryos are a distant prospect, but they raise ethical questions about the definition of life and the potential for creating organisms entirely in the lab. The road ahead, promise and peril. These advancements, whether enabling same-sex couples to have biological children, reversing aging, or merging minds with machines, share a common thread. They challenge the limits of biology and ethics. The O chromosome concept, while speculative, reflects the trajectory of IVG, which could democratize parenthood, but risks genetic unintended consequences. Age reversal in mice hints at a future where aging is a treatable condition, yet human applications are fraught with complexity. CRISPR, BCIs, and synthetic embryos further blur the line between science fiction and reality, demanding careful oversight. As these technologies progress, society must grapple with profound questions. Who gets access to these innovations? How do we balance individual desires with collective risks? And where do we draw the line between healing and enhancement? Scientists, ethicists, and policymakers will need to collaborate to ensure these breakthroughs benefit humanity without opening Pandora's box. For now, these discoveries remind us of science's power to surprise and inspire. From an accidental anti-aging drug to the possibility of 2 dads sharing a genetic legacy, the future of biology is as thrilling as it is uncertain. Sources: in vitro gametogenesis and reproductive technologies. Age reversal in mice. Note: specific study details require further primary source verification. CRISPR and gene editing. Brain-computer interfaces and synthetic embryos. Note: Neuralink updates based on recent reports. Note: the O chromosome concept appears to be a hypothetical or emerging idea not yet standardized in scientific literature. This article interprets it within the context of IVG and related technologies. For precise details on unpublished or speculative claims, further research is needed. By Scott Young, aka Synfamous Gamer.